Welcome to G Fun Facts Online. For decades, the war on cancer has been fought on one primary battlefield: proteins. These are the workhorses of our cells, and when they go rogue due to genetic mutations, they drive cancer's growth. So we've designed drugs to block them, inhibit them, and shut them down. But what if we could stop the war before the soldiers even reach the field? What if we could intercept the orders? A new frontier is opening in molecular oncology, one that targets cancer's master blueprint, its ribonucleic acid, or RNA. This is the world of smart drugs, and it's promising to rewrite the playbook for cancer treatment. To understand this revolutionary shift, we need a quick biology refresher. Remember the central dogma? DNA holds the master blueprint for life. To build anything, like a protein, the cell makes a temporary disposable copy of the relevant DNA instructions. That copy is called messenger RNA, or mRNA. This mRNA then travels to the cell's protein-making factories. So the flow is: DNA makes RNA, and RNA makes protein. For years, we've focused on the last step, stopping the proteins. The problem is, it's estimated that only a small fraction of all disease-related proteins are druggable. Many of the proteins that drive cancer are like ghosts. They lack a well-defined physical pocket for a drug to latch onto. But RNA is different. It's a physical strand of code. And if you can target the code, you can stop the harmful protein from ever being made. You're not fighting the enemy's soldiers. You're cutting their communication lines. This opens up a vast new landscape of targets that were previously untouchable. So how do we do it? Scientists have developed an entire arsenal of RNA-targeted weapons. First up are the gene silencing pioneers, antisense oligonucleotides, or ASOs. Think of these as molecular sharpshooters. They are short, synthetic, single strands of DNA or RNA, designed to be a perfect mirror image of a specific cancer-causing mRNA sequence. When the ASO finds its target inside a cancer cell, it latches on through simple base pairing. This binding event can trigger a few different outcomes. Most commonly, it flags the mRNA for destruction. The ASO-mRNA hybrid attracts an enzyme called RNase-H, a cellular demolition expert that sees this strange structure and immediately shreds the mRNA. No mRNA, no protein. ASOs can also act as a physical roadblock, literally sitting on the mRNA and preventing the cell's machinery from reading it. In the clinic, ASOs have shown both promise and challenges. For example, an ASO called AZD4785, which targets the infamous mutant KRAS gene, has shown potential in preclinical models of lung cancer, offering hope against a notoriously difficult target. Next, we have small interfering RNAs, or siRNAs. These molecules harness a natural process that already exists in our cells, a defense mechanism called RNA interference, or RNAi. An siRNA is a short, double-stranded piece of RNA. When it gets into a cell, it's loaded into a protein complex called RISC, which stands for RNA-induced silencing complex. The RISC complex then unwinds the siRNA and uses one of its strands as a guide. It patrols the cell, searching for any messenger RNA that perfectly matches that guide strand. Once it finds its target, a molecular scissor inside RISC, an enzyme named ARGONAUTE2, snips the target mRNA, leading to its degradation. The advantage of siRNAs is their incredible potency. A tiny amount can trigger a powerful and long-lasting gene silencing effect. A great example in development is Sig12D loader. This is a tiny biodegradable implant loaded with an siRNA designed to shut down the G12D mutated KRAS oncogene, a key driver in many pancreatic cancers. It's currently being tested in patients with locally advanced pancreatic cancer, delivering the therapy right where it's needed. Now, let's talk about a technology you've almost certainly heard of: mRNA vaccines. Their success against COVID-19 was a watershed moment. And now that same technology is being aimed squarely at cancer. But here's the twist. Unlike ASOs and siRNAs that directly attack the cancer cell's machinery, mRNA cancer vaccines work by training your own immune system to do the job. Here's how. The vaccine delivers mRNA instructions for making a protein that's found on the surface of the cancer cells, known as a tumor antigen. Your own cells take up this mRNA, produce the antigen, and then present it to your immune cells, essentially holding up a most wanted poster. This trains your T cells to recognize and hunt down any cell in your body displaying that specific antigen, namely the cancer cells. The results from recent clinical trials have been nothing short of stunning. A landmark Phase 2 trial tested a personalized mRNA vaccine, mRNA 4157, for patients with high-risk melanoma. Each vaccine was custom-made based on the unique mutations in the patient's own tumor. When combined with an existing immunotherapy drug, this personalized vaccine reduced the risk of cancer recurrence or death by 49% after 3 years. That is a massive leap forward. With over 120 clinical trials for mRNA cancer vaccines underway for various cancers, this is one of the most exciting areas in all of medicine today. Finally, in our arsenal, we have RNA-aptamers. If ASOs and siRNAs are the warheads, think of aptamers as the guidance systems. These are short, single-stranded RNA or DNA molecules that can fold into unique 3D shapes. This allows them to bind to specific targets, like proteins on the surface of a cancer cell, with incredible precision, much like an antibody. In oncology, aptamers are being developed as chemical antibodies for targeted drug delivery. You can attach a chemotherapy drug or an siRNA to an aptamer that only recognizes cancer cells. The aptamer then acts as a molecular GPS, delivering its toxic payload directly to the tumor while sparing healthy tissues. For example, an aptamer called AS1411, which targets a protein called nucleolin that is common on cancer cells, has been tested in trials for kidney cancer, showing excellent tumor targeting with low toxicity. All these technologies sound amazing, but they all face a common major hurdle: delivery. Naked RNA is incredibly fragile. Enzymes in our bloodstream would shred it in minutes. And even if it survived, its negative charge makes it hard to cross the cell's oily membrane. This is where the smart drugs really comes into play. The most common solution is nanoparticles, specifically lipid nanoparticles or LNPs. If you received an mRNA COVID vaccine, you've already experienced LNP technology. These are microscopic bubbles of fat that encapsulate the delicate RNA, protecting it on its journey through the body and helping it fuse with the target cell to release its cargo. But it gets even smarter. Researchers are now designing stimuli-responsive nanoparticles. These are carriers designed to break down and release their drug only in the unique environment of a tumor. For instance, the area around a tumor is often slightly more acidic. A smart nanoparticle can be built to dissolve in that acid, releasing its payload with precision. Others are designed to break apart only in the presence of specific enzymes that are overproduced by cancer cells. A particularly exciting new technology is the development of RIBOTACs, which stands for ribonuclease targeting chimeras. These are small, two-sided molecules. One end is designed to selectively bind to a specific disease-causing RNA. The other end acts like a powerful magnet, recruiting one of the cell's own naturally occurring RNA-destroying enzymes, called RNase L, directly to the target. Once recruited, RNase L obliterates the RNA. This is a game changer for targeting RNA structures that were previously considered undruggable. A recent study showed a RIBOTAC could destroy a long, non-coding RNA called TERA, which helps make some cancer cells immortal, opening the door to a whole new class of precision medicines. Of course, for any of this to work, we have to solve two other problems: stability and specificity. We've already discussed how nanoparticles help with delivery, but the RNA molecules themselves are also chemically modified. Scientists can swap out certain atoms in the RNA's backbone or sugar, making them resistant to degradation and helping them bind more tightly to their targets. This is what turns a fragile lab molecule into a robust drug. Specificity is about avoiding off-target effects, making sure your gene-silencing drug only silences the gene you want it to. Researchers now use sophisticated computer algorithms to design siRNAs that have minimal risk of accidentally matching other, healthy genes. The ultimate goal of all this work is truly personalized medicine. Using a technology called RNA sequencing, or RNA-seq, doctors can create a complete, dynamic snapshot of all the RNA molecules in a patient's tumor. This tells them exactly which genes are active and driving the cancer at that very moment. Sometimes RNA-seq can find targets that DNA testing alone would miss. For example, at Columbia University, the Oncology platform uses a patient's tumor RNA profile to identify the master regulator proteins driving their cancer. It then uses a computer model to predict which drugs will be most effective at shutting down those specific master regulators. A clinical trial for advanced pancreatic cancer patients based on this approach is currently underway. And the future is getting even more precise. You've heard of the gene editing tool CRISPR, which cuts DNA. Well, there's a new version called CRISPR-Cas13 that specifically targets and cuts RNA. The huge advantage here is that the changes are temporary. You are not making a permanent, potentially risky change to the cell's genome. You're just editing the disposable mRNA messages. This could be used to temporarily dial down a cancer-causing gene, or even correct a disease-causing mutation in the RNA itself. The road ahead isn't without its challenges. Developing these therapies is incredibly expensive. Manufacturing them at scale is complex, and the regulatory path for such a new class of drugs is still being paved. But the pace of innovation is breathtaking. The most likely future of cancer treatment lies in smart combinations. Perhaps an mRNA vaccine to prime the immune system, combined with an siRNA that silences a gene the cancer uses to hide from that same immune system. We are witnessing a pivotal moment in oncology. We are moving beyond fighting the downstream effects of a cancer's genetic mistakes, the proteins, to intercepting the messages themselves. This is more than just a new set of drugs. It's a fundamental shift in how we understand, target, and treat cancer, rewriting its very code to, ultimately, save lives.